let's get started. Couple of announcements before we do. The first one is this. Next term, if you're looking for what I think is going to be a hot class, maybe, mutant viruses from hell, also known as the Advanced Molecular and Cell Biology Research Lab. This is a course where you try and figure out what's going on with this virus, which is the main focus of what we're working on in my lab. Basically, what happens is day one, you get a couple of mutants, and we have no idea what they are, or whether they work or whether they don't. And then in the next 10 weeks, you figure out and tell us whether they work, where the mutants are, et cetera. So yeah, I can tell you in about 30 seconds what happens. It takes 10 weeks versus labs. Monday, Wednesday, and Tuesday, Thursday sections, 1 to 3:30 in the afternoon. Any questions about that, please let me know. Also known as a course-based undergraduate research experience, which I think over here is much more what it's about. So that was my first announcement. My second announcement is, as I mentioned before, our departmental seminar, which is particularly important this week because we have a candidate for a position in the department, Dr. Brooke Napier from Stanford, who is going to be talking about immunity and sepsis, particularly having to do with the extracellular part of E. coli and a lot of other gram-negative bacteria, particularly the lipopolysaccharide, which is involved in that process. So it should be a lot of fun. That's tomorrow at noon. Of course, it doesn't say on here, which we need to check that out. So this Thursday at noon in room 247 in SRTC, which is literally right across the hall from the biology office. And then our final speaker, or I should say final candidate, is going to be coming at the beginning of March, and I will make sure to remind you when that is happening. Last announcement, of course, is that on Friday, we'll have a couple of visitors, both the dean and I think it's now 4 people from the biology, teaching of biology course who'll be frantically taking notes about all the obnoxious questions that I ask you and vice versa. So, and then, of course, a week from Friday is, again, another midterm. How did that happen? And we're already halfway through the term. Go figure. How did that actually work? So I have a couple of sort of follow-up slides from last time in terms of what this whole mess looks like in terms of the various different parts of the spliceosome. So this is a paper from last year in Nature where they look at the structure of what they call the tri-SNRNP. So SN, small nuclear ribonuclear protein complex, U4, U6, U5. U4 and U6 are in the gray and blue in here, and then U5 looks to me exactly the same color blue down here at the bottom. It's actually a lot easier to see and look at in a slightly earlier paper that these guys published, which is where they basically gray out all the proteins, and then you can look at the RNAs. And so here, U4 is base pairing with U6, and U5 is basically down here base pairing more or less with itself. And then lots of other proteins that are involved in this process. This tri-SNRNP, that's the SNRNP complex that comes into the spliceosome after you have U1 and U2 associated with the 5' end of the intron and the 3' end of the intron. And the other thing that a lot of people ask me about after class is, well, what's going on with the RNA helicases, and why doesn't U6 automatically have activity when it's just sort of floating around inside the cytoplasm? And the reason for that is that it's heavily base paired. And so I think this is a nice example right here. U6 is base paired with U4. And basically what that does is it changes the structure of U6 which means it does what? Changes the function of U6. So you basically need to get rid of all these base pairs with U4 to allow U6 to form different base pairs, starting with the 5' end of the intron, and then allowing it to make the active site where it actually catalyzes the splicing reaction. So hopefully this is a little more clear, maybe less so. Yeah. Sure. So what is a helicase separating? So the helicase is taking double-stranded, in this case RNA, and making it single-stranded. So it's pulling the two strands apart, just like what happens in terms of the helicase in terms of DNA replication and the helicase activity that you have with the RNA polymerase, pulling those two strands apart. And so here, what it's going to be doing, I'll get back to a sec, is it's pulling these strands apart, these two that are bound to each other. Turns out it's also involved in removing the double-stranded pieces that you have here. Yeah?
Now, since it's separated, doesn't it have what keeps it from coming back together?
So what keeps it from coming back together is the rearrangements which are happening in the process. There are multiple different ways. If you just think about base pairing, all you need to have is complementary bases. And so there are lots of different ways that it could bind. So once the helicase has happened, now you'll have other conformational changes that happen which allow it to base pair somewhere else. Yeah?
The HNRNPs, the book says that one of their purposes is to unwind the hairpin helices in RNA. So are the helicases part of the HNRNPs, or is this another process that's going on with the HNRNPs separate from helicase activity?
Okay, so the question basically has to do with HNRNPs. What the heck are they doing, and can they be serving as helicases in this particular case? The problem with HNRNPs is the H, heterogeneous, so it's a whole bunch of different proteins that we could try and get into huge amounts of details on. Some of them are helicases. They don't actually seem to be required for the actual splicing activity. You can purify the spliceosomal components and put them in with an appropriate piece of exon and intron without any of these HNRNPs and still get splicing to take place.
So is this just for the cellular processing of the lariat post splicing? Is that what they're there for then?
Oh, okay, so paraphrase again here. It's okay. Yeah, right. So why are HNRNPs there? Never ask why questions in biology is the answer to that question. The answer is we really don't know. Probably they're involved, and again, this is probable and hand waving, which is the best we can do with any of these why questions. Probably involved in making sure that the intron is an intron and it gets recognized as being an intron, and that in that particular case, in those introns, when we talk about nuclear export as well, you don't want to have those HNRNPs associated with the RNA, because if they still are associated with the RNA, that's kind of a red flag that tells the rest of the system, again, to totally over-anthropomorphize, this guy hasn't been spliced yet. Don't export it. Don't take it out. Don't try and get it translated. So as far as we're concerned, HNRNPs are basically identification of introns. Again, identification of introns, that it's a specific intron. There are way many other things, but why? Anybody's guess. Ask Darwin. So that's all I wanted to do, talk about actual mechanisms. Oh, sorry.
The SNRNAs for
Small nuclear RNAs. SNRNA stands for small nuclear RNA, and SNRNP is small nuclear ribonucleoprotein complex. So this, for instance, is SNRNP. So it's got RNAs and it has proteins, and it's in the nucleus because it's doing this job. Okay, other questions on splicing mechanisms? Okay, so we'll talk a little bit more about splicing. We want to talk about alternative splicing, and then move to the 3' end of your messenger RNA, talk about polyadenylation, and this also, as I mentioned before, ties into what we think is going on with transcriptional termination. So a lot of this is a lot of modeling at this point, still a lot of new data on it. Then we'll talk a little bit about RNA export, which is getting back to the hnRNPs, and then talk about ribosomal RNA, and particularly modifications of ribosomal RNA. What polymerase makes ribosomal RNA in eukaryotic cells? RNA polymerase 1, 2, or 3? Ribosomal, 1 and actually a little bit of 3 too. So not RNA polymerase 2. So we can ignore RNA polymerase 1 and 3 for the time being, but then we'll get back to that. Those transcripts that are made by RNA polymerase 1 and 3 don't usually get spliced. Why? So 1 makes most of the ribosomal RNAs, 3 makes one of the smaller ribosomal RNAs. So why don't they normally splice? Where are all the splicing proteins and things associated with the polymerase? The CTD, does RNA polymerase 1 and RNA polymerase 3 have CTDs? No. So they're not going to have the proteins that you need. They're not going to have U2AF. They're not going to have the branch binding protein, etc. So with Pol II transcripts, so the messenger RNAs, these do get spliced. And it turns out that about 60% of genes in humans, so these primary transcripts that are made, get spliced in different ways in different cells at different times. So alternative splicing, i.e. which exons you use, which exons you don't use, really is highly variable. So when I talk about 20,000 protein coding genes in the human genome, just one of these genes, so for instance here the alpha tropomyosin gene, that's a one gene, but in different cell types, and even sometimes in the same cell type, you can have different splicing that takes place, and that'll give you different exons, which is going to end up giving you a different protein. A lot of it's the same. Almost always the 5' end of your messenger RNA is going to stay the same. So the N-terminus of your protein is going to be the same. But then you're going to have lots of modifications through the rest of this primary transcript as it gets spliced. And my favorite example of this is from the fruit fly, Drosophila, where there's one particular gene that can get spliced in 38,000 different ways. So yeah, maybe relatively small number of genes, but a ridiculously large number of slightly different proteins that can be made from any of those things. And the way that you can actually do the calculation here is each of these colored regions is an alternative exon. One and only one of the exons in each of those groups get used. So 12 possibilities times 48 possibilities times 33 possibilities times 2 possibilities, you end up with 38,016. This is extreme, but it gives you a bit of an idea that alternative splicing can give you a whole bunch of different proteins from one particular gene. So how is this regulated? We talked a little bit about this last time, having to do with nucleosomes and the fact that splicing happens as transcription is happening. So if you're transcribing your way along the DNA, the RNA is being produced, the splicing proteins jump onto that RNA from the CTD and then start doing their splicing job. So that means that the exons at the beginning of your RNA are much more likely to be spliced to each other rather than one at the beginning to way down at the end, because it just hasn't been produced yet. So there's no way you can actually do that particular splicing reaction. So splicing happens in a mostly linear fashion, going from your 5' end to your 3' end. Turns out that there's some really nice experiments which show that if you go really fast in terms of transcription, and the way to do this is get rid of all the nucleosomes, then you will end up doing a lot more splicing to different places in your primary transcript. On the other hand, there are also some histone modifications that can slow down, or in some cases also speed up the transcription, and even some of them are going to associate with some of the splicing factors, which as we'll see in just a second, are particularly important for some of these alternative splicings. Yes?
Does that prevent you from using the same exon twice for the same protein? If it's linear, it progresses in a linear fashion?
I'm not sure I quite understand your question. You may restate it?
If you have 5 exons
Yes.
in a string of RNA,
Yep.
in that what I'm asking is whether or not you can use that first exon twice. So
Can you use it twice?
have to be duplicated
Yeah, so just the fact that it's an exon, all splicing is going to be subtractive. It's taking parts out of that first RNA which you've made. So that exon can only be used once. Now, it could be varied in terms of length and where it is, but in terms of the splicing, it's all going to be reductive with that one little tiny exception. We have trans-splicing that takes place, but we're going to ignore that.
So, are you saying, you said that when you move faster, if you take all the nucleosomes off, you get different splicing?
Yes.
Are you talking about the variety of splicing gets larger, or the amount of alternative splicing?
So basically what happens when you speed things up is you're going to start skipping exons. So and we'll talk about that just here in just a second, how that's happening. And if you slow things down, you're more likely to use all of the exons. And that's been quite nicely shown. So what determines this? Particularly if we're talking about alternative splicing like that DSCAM1 in Drosophila, where you've got all kinds of different possibilities in terms of what exon actually gets used. Turns out that which exon gets used really depends on, not terribly surprising, where you have U2 associated with the intron. U1, always going to be associated with your exon right at the end. You remember the base pairing interactions that we saw there. U2 has to get associated, usually very close to the 3' end of your intron, also where this branch is going to form, usually through interactions with U2AF. How do you get U2AF bound to these exons is you very often have these, we talked about them last time as being SR proteins, but why are they called SR proteins? You've got lots of arginines and serines, and put them the right way around, it's arginines and serines, opposed to serines and arginines, which is what we talked about last time. So these are just the same proteins that we were talking about before. So SR and RS, exactly the same thing. These proteins, the RS proteins in particular, bind to exons, and this is very much in a cell type specific or developmental time specific way. So these are specific RNA binding proteins that will bind to exons. If the SR-RS protein binds to the exon through these little nice little arrows here, interacts with U2AF, helps U2AF bind. U2AF is our factor which is going to help U2 to bind. Now, what they're binding to here is the exon, but it's a specific part of that exon. And those exons that are binding to these SR proteins are called enhancers. Enhancer means a DNA, nucleic acid, I should say. We'll talk much more about enhancers after the midterm. A nucleic acid sequence that proteins are going to bind to. So SR proteins bind to these splicing enhancers, stimulate U2AF binding, getting U2. If U2 is here, it's going to interact with U1. We'll get nice lariat formation, and splicing will happen. If, on the other hand, you have these pink proteins, the RG proteins, again, cell type specific, developmental type specific, which are going to bind to now silencers, again, specific nucleotide sequences the proteins are going to bind to, they will not stimulate binding by U2AF. And so this whole complex will, as we're transcribing along here, and again, faster transcription, you're going to get to this next exon sooner, that will have a U2AF and a U2 associated with it, and just ends up making a bigger intron and chopping out this exon completely. So this is how those processes occur, and it's really very much about where U2 is going to be associated with the RNA. So this gives you a whole bunch of possible ways that things can happen. And we just outlined this version up here at the top. So if you've got SR proteins that are bound to this particular exon, you'll include it. If you have GR proteins that are associated with there, it's not going to be used. But that's only one of a whole bunch of different possibilities here. We're not going to go into all the details on here, but all of these are known different kinds of splicing that can take place. You could start in the middle of what otherwise is an exon. You could splice into the middle of what's otherwise an exon. In some cases, introns are actually not cut out at all. In a lot of cases, and this is also true for that DSCAM1 gene, if one exon is included, the next one is not included in those sets of potential exons which are there. And even in some cases, you'll have a different piece of RNA. Sometimes if the promoter is here versus over here, the promoter here is going to give you these two exons, but if the promoter is right here, then you end up with different exons which are being made. So where the promoter is for any particular gene can also determine what you have in terms of that. And then also alternative poly-A sites where you end up with termination of transcription can be determined partly, again, because we're having this concurrent capping, splicing, termination process take place. Sure.
So, say on the first one, like I said, alternative exons.
Yep.
If you start at that first yellow sequence.
This one here. Start at the first yellow sequence. Yep.
Then you splice those two first ones together.
Two first ones are always spliced together. Yes.
If you start after the second one,
Here.
you have two possible alternate outcomes.
Yeah. Well, this is going to depend. Is if this exon actually gets used as you're being transcribed, it might or might not. Now this is here. We've got transcription starting over here, and your primary transcript has everything in here. And then in some cases, you'll have splicing between these two exons. And in some cases, you have splicing with this exon and to this exon. The same gene, the same DNA, in some cases is going to give you one splicing outcome, and in other cases will give you a different splicing outcome. These are different possibilities, exactly. Yeah. What happens to the intron which doesn't get taken out in this case right here? You know, wherever it is, down here. You know, if my pointer would work, that would help. This one right here. It turns out that these are actually even sometimes coding sequences and maybe regulatory sequences as well. So, but why you call it an intron versus an exon and retained is really kind of a bizarre question. Usually what happens in this case is these end up being what are called upstream ORFs, or open reading frames, that we'll talk a lot more about as we move forward in the class. But it's actually one of those very rare cases in eukaryotic cells where you have two different proteins coded for on one RNA. So a polycistronic RNA. Very common in bacteria, very rare in eukaryotic cells. Yeah?
I was just wondering if that included exon or intron can also serve as domain holders for base barrels.
Yeah, if the intron sequence is actually coding, it could be. But then, since it's still by definition here an included intron, it's probably not going to be a coding sequence. Okay, why are we spending so much time talking about this? All these alternatives? Well, because 60% of our genes are alternatively spliced, but also a number of genetic diseases are quite well known as being splicing problems. And so many single nucleotide substitutions, particularly if they're right at the beginning or right at the end of your exon, where you've got those conserved nucleotides, completely conserved nucleotides. Those can mess up splicing in such a way that the gene which is produced is no longer functional or misfunctional. And one of these is very well studied is beta thalassemia, because it's looking at beta globins. So beta globin's part of the hemoglobin. Normally, and this is just a part of the gene, you have 3 exons that get put together. They're relatively close to each other. We actually looked at that as the example of not terribly heavily spliced before. But if you have a single mutation, for instance here, in those conserved nucleotides that are right at the end of the intron, you're not going to have U2 binding here, right? So U2 doesn't bind there. It's going to bind over here. So you miss this second exon. And a number of people have these particular mutations. Beta thalassemia is actually not a lethal mutation. So you have patients who have these things. Single nucleotide changes in this splice, what they say, splice acceptor site, or the 3' end, would be normally the beginning of your exon. If you have a mutation there, the splicing is still trying to happen, and it happens at a suboptimal location, adds this extra sequence right here, which it turns out doesn't code for the right amino acids and messes up the structure again of your beta globin gene. And in one case, which is a very rare case, still been found, these single point mutations here can lead to the incorporation of an extra exon. So instead of having 3 exons, you end up with 4 exons in terms of the final coding. And these are literally, B, C, and D are all specific mutations which are known to happen in people with beta thalassemia. In fact, one of my master's students has beta thalassemia and hates to go to the doctor because they're like, oh, wow, really cool. Yeah. It's like, no. She's now a professor of veterinary medicine, so she's doing okay. But these are kinds of changes, and particularly genetic changes, very often these spontaneous changes. It's a single nucleotide change. And how often the single nucleotide changes happen and escape the whole mismatch repair machinery? About 1 in a billion replicated nucleotides. So we got about 6 billion nucleotides, so every time we're going through and replicating, there'll be 6 changes. So these single nucleotide changes can actually have some pretty big effects, and we've seen them. Okay, that's all we have with splicing except for clicker questions. Exactly. So, presence of which of the following factors in a specific cell is thought to determine whether an exon is used or not in alternative splicing? BBP, SR proteins, 5' splice donor, 3' splice acceptor, U4, U5, U6. Ten, 5, 0. One of these days, I'm still waiting for it, we're going to have 100%. But we're not quite there yet. And probably one or the other of you is going to try and spite me all the time and answer the wrong one. But yeah, so there are a couple of things that are important here. One is the specific cell. So you've got branch binding protein, donors and acceptors, and U4, U5, U6 in all cells. So just a specific cell basically has already eliminated 4 of them. So even if you didn't know that SR are S proteins, those that have serines and arginines that are quite common in them, are going to be determining the alternative splicing. So yes, now the scary thing would be if 95% of the people had gotten it wrong. That means I would have been doing a really bad process of describing this. So I wanted to spend a couple of minutes talking about, oh, sorry. Oh, what's BBP? What's BBP? Branch binding protein. So that's going to bind to that A residue, which is going to provide your 2', 5' phosphodiester bond. Okay, so one of the questions that I think somebody asked after class actually last time was, where do introns come from? Why is it so ridiculously inefficient? And again, one of those why questions. Never ask why questions. But one thought is because you have 60% of these differential splicing that can take place, that means you've got a lot more variability in terms of different proteins that you can get, tweak your activity a little bit. So probably why is an evolutionary kind of thing. But then the next question, which again you should never ask in biology, is where did it come from? And that we actually have some pretty good evidence for where splicing came from originally. And that comes from the so-called self-splicing introns. So a number of researchers, particularly Tom Cech in the late 80s, early 90s, was apparently doing some experiments looking at some RNAs that they purified. And they're actually looking for RNA binding proteins and trying to see how these RNA binding proteins were interacting with each other and how they were causing splicing. And then rumor has it, one day there was a graduate student or postdoc, came in and talked to Tom Cech and said, I've purified this thing as well as I can and it's still splicing. And it turned out, make a long story short, it was literally the RNA itself which was doing the splicing. RNA in the absence of any protein whatsoever. And this was the so-called group 1 self-splicing intron. Turns out it's a very specific sequence, which of course is going to base pair and give you a very specific structure, which will then serve to literally splice out this particular sequence from your RNA. Later on, a second type, again what's called group 2, was found, self-splicing intron sequence. Turns out that this also has a conserved adenine residue, a 2' OH, forms a lariat structure very much like what we see in terms of the spliceosome right now. Now, this particular self-splicing intron depends very much on the exact sequence which is present in the intron right here, because it's got to fold up on itself. It's basically got to provide all of those extra specificity pieces, the activity pieces, etc., in terms of all these base pairing interactions. So that's not a very good way of giving yourself lots of flexibility in terms of how splicing could happen. So what seems to have happened, again over an evolutionary timescale in eukaryotes, is that basically these sequences were split up into the 5 different SN RNAs that we have and associated with proteins in order to make this a much more flexible kind of process. But the other thing that came out of this, which was the huge surprise and why Tom Cech got the Nobel Prize, was that RNA itself had enzymatic activity. And this was the first time that it was actually shown was in these self-splicing introns, that the RNA just by itself had enzymatic activity, which is been taken as really strong evidence for what people also call the RNA world. So when life first arose on this planet or maybe some other planet and came here later, we can talk more about that some other time, that RNA was being used as both the catalytic molecule for all biochemical reactions, but also at the same time carrying the genetic information. So RNA can do both of these things. And RNA does a much better job of that than DNA does. And we have, if we have a chance, after we're done with the translation stuff next Wednesday, we may have a chance to talk a little bit more about, next Monday, we have a chance to talk a little bit more about some of the RNA world and how some of these ribozymes may have been important very early on. So now I want to switch gears a little bit, talk about that 3' end of our messenger RNA. We all know that it ends up with a whole bunch of adenines at the end, the so-called poly A tail, about 250 of these for most human messenger RNAs, somewhat shorter, somewhat longer in some of the other eukaryotes. How do you get that? You have a primary transcript, again, shown just up here, and a couple of sequences that are present in this nucleotide transcript. I don't expect you to remember sequences here. Where, not surprisingly, you're going to have proteins that associate with this RNA, and then there's an endonuclease, which is going to cut in the middle of this RNA, gives you a 3'OH, and then there's a addition by the so-called poly A polymerase, again, incredibly creatively named, which adds a bunch of adenines to the end of this now final messenger RNA. So let's look at this in a little bit more detail. There are a couple of proteins that I expect you to remember, conveniently labeled here in red. CPSF, the cleavage and polyadenylation specificity factor. The cleavage stimulation factor, CSTF. Again, I didn't come up with these names. Poly A polymerase, PAP, and the poly A binding protein. So how does this process work? Transcribing, RNA polymerase is happily transcribing along the DNA, gets to this sequence, which is going to encode this sequence that the cleavage and polyadenylation specificity factor associates with. So basically what this does is says, hey, we're almost at the end of where this transcript should be. This is, of course, conveniently carried along in the tool belt, hanging off of RNA polymerase II, that C-terminal domain. Turns out originally, how does it get there in the first place? TF2D actually associates with CPSF to get that particular protein to the pre-initiation complex before you have transcriptional initiation. Same thing is true with CSTF. So these guys are put onto the C-terminal domain. Once you have the right binding site for CPSF, that will bind cleavage stimulation factor, lots of additional cleavage factors, which we're not going to talk about here, including the endonuclease, which cuts in the RNA. Once that's been cut, now you have a 3'OH that the poly A polymerase can bind to and extend. This is an RNA polymerase which is not template dependent. It just will add adenine residues. And as it adds adenine residues, the poly A binding protein will start to bind to the poly A. And eventually, what seems to happen is this poly A binding protein, once it gets to the end, it kind of bangs off the poly A polymerase. So you end up with approximately 250, again, in the case of human cells that you have here. So your final messenger RNA has all of these poly A binding proteins bound to the end here, just after the binding site that you had for CPSF in the beginning. So now the question is, what happens with what's left? Because that cleavage, the RNA polymerase is still continuing to transcribe. And so this, unfortunately, they didn't have an image in our textbook, so I went and pulled one up from alternative textbook. That's shown here. So again, our RNA polymerase here with its C-terminal domain has CSTF, CPSF bound to it, gets to this site, cleavage takes place, poly A tails happen, but now you have a 5' monophosphate and no cap at the end of this particular RNA. So now there's a exonuclease. We've talked quite a bit actually. These are the ones that chew in at the ends of nucleotide chains. which basically chops up the RNA as it's being made by the RNA polymerase, and it gets to a certain point here, and what we think happens, and again, this is a lot of hand-waving here, is that that exonuclease actually pulls the RNA out of this elongating polymerase in a manner quite similar to what we think happens with these hairpin loop structures and the poly-U sequences. So structure which pulls the RNA out of the RNA polymerase. Once that RNA has been pulled out of the RNA polymerase changes its structure, falls off of the RNA. Yeah? So the question is, where's CFTF and the CPSF? So they're actually all still associated with this RNA. So this RNA is now separate from the polymerase, this bottom RNA here. So this light green thing here, that's going to be where you have binding by the cleavage and polyadenylation specificity factor, so CPSF and CSTF. They're all going to be bound here. And this end will be released. This piece of RNA is now floating away. It's a separate thing. Now the poly-A polymerase is associated with it. Yeah, I'll get back to you in a second.
The book describes it as like the GU-rich area is where you cleave off. And that gets left off and basically replaced with the poly-A tail. So the GU-rich area, and that's where the book describes CSTF binding space. So if you cleave that off, does it jump onto the AU part, or does it remain
where CSTF on the stimulation factor? This, again, this is why, additional cleavage factors. So all of these additional cleavage factors are going to displace most of these proteins, which are here, and end up giving you the poly-A polymerase. So originally, yes, CSTF is going to bind there, but by the time you've got the poly-A polymerase and the cleavage taking place, it's either no longer there, it gets bumped off by the exonucleases it's going through.
So yeah, once it's off, once that transcript is off and the poly-A tail is added, CSTF is no longer.
Well, it may get bumped off, it may still be associated with it, it may fall off. Again, I don't know all the details on that. It's probably known, I just don't know what they are. Yeah? Yeah, so you're exactly right. The second part is right. So it's the poly-A polymerase which is adding the poly-A. It has nothing to do with RNA polymerase II. RNA polymerase II, again, to totally over-anthropomorphize here, has no clue what's happened to the RNA which is just made. It's just blindly chugging along here, and until the RNA gets pulled out of it, from behind. Really, not fair in terms of that process. So yeah. Okay, so the question here, are these two separate theories for termination, or are they happening in conjunction? Maybe I should have tried to make this a little bit clearer. This is what's going on with RNA polymerase II. This is the bacterial situation down here. So completely different. Okay, and here, this is clearly a very sequence-specific place that your termination takes place, whereas here, it just kind of depends how fast your exonuclease is going and how fast your polymerase is going, where you actually end up stopping. Okay, just to finish up with this, I'm hopefully not, again, beating the dead horse in this particular case. Almost all of this splicing, capping, termination, etc., has to do with all the proteins that are hanging off of this CTD. And I like this figure because it actually gives a much better indication on how big the CTD is relative to the rest of the RNA polymerase. It's pretty huge. And it's this repeated 7 amino acid sequence with lots of phosphorylation on it, and the capping enzymes are associated with it, the spliceosome enzymes are associated with it, the cleavage and polyadenylation factors are associated with it, etc. And then, just because they're close to where this RNA is, they can clearly associate with that relatively rapidly as it's being formed. And there's a couple of pieces of evidence for this. People have shown quite nicely that this cleavage and polyadenylation specificity factor interacts directly with U2 snRNP. So there are interactions between splicing and capping here, and splicing and tailing proteins, which are here. So one thing that, sorry, yeah. Let's ignore the transcription elongation factors for now. That has nothing to do with poly-A tail. It has to do with the elongation and how the polymerase is moving along the DNA. So then is it the polyadenylation that's adding the poly-A tail? Yes, the cleavage and polyadenylation factors here, which are adding the poly-A tail. And that includes the poly-A polymerase. So one question that has been asked quite a bit, we're all looking at single pieces of DNA with all of these transcription factories running along it, etc. What's really happening inside the cell? Well, what actually happens inside the cell, and of course this is inside the nucleus of the cell, it turns out that it's way easier to move the DNA than it is to move all these proteins around. Because if you think about a cell, it's actually really kind of crammed together, all of the macromolecules, etc. And the DNA or the RNA is relatively small compared to all of these protein complexes. And think about the spliceosome that I showed right at the beginning. This is a ginormous thing with all of these extra pieces on it. So it actually seems to make sense that you have all of these things located in one particular place in the nucleus. And if you remember when we talked about nuclear structure, way back when, before the first midterm, we've always forgotten that stuff. If you look at transcribed genes, where they're in the nucleus, they move. They move relative to where they normally are when they're not being transcribed. And that movement seems to be to some of these, what people also call a transcription factory. And they're specific areas. There's not just one inside the nucleus. There are quite a few of them. But they're not spread out all over the place. And so that's just what's shown right here, is that these are all really associated with each other. So it seems as if all transcription, RNA modification is happening in very specific places in the nucleus rather than your transcription factory, kind of like what we saw in the animation where it's sitting there and then boom, takes off along the RNA. It's really much more concentrated in one place. So just wanted to mention that again. Everything we talk about in this class is ridiculous oversimplification. So we've made RNA. I'll just get to it in a sec. We've done the capping, we've done the splicing, we've done the tailing. This is all happening in the nucleus. And all translation is going to be happening out in the cytoplasm. So we got to get it from the nucleus to the cytoplasm. Yeah, sorry. Your question. Okay, so the question is basically about order to some extent. So if you just think about how it's being transcribed, you're going to have the cleavage and polyadenylation specificity factor that binds first, because that's going to be the first piece that comes out of the RNA polymerase. Once that's bound there, then it brings in the cleavage stimulation factor, which will bind in its GU-rich sequence, and that will then start assembling all of the extra polyadenylation stuff. So it's first the cleavage and polyadenylation specificity factor, and then everything else. But it makes sense if you just think about how the RNA is being made. Okay, so we've made our RNA. Now it needs to get out. And there are a couple of important things about getting out. One of them is what we talked about before, these hnRNPs. An hnRNP, if it's still associated with your RNA, what does that mean? It means splicing hasn't happened, or it hasn't happened properly. So that's kind of a red flag and says, hey, this is not a good RNA. It's not done yet. It hasn't been, in the oven long enough. So a couple of other things. SR proteins. SR proteins are associated with what? Associated with exons. And so those associated with the RNA are potentially a really good way of saying, hey, those are associated with RNA. We know that that exon has been used. But there are also these, they have them here. I think I may have neglected to mention before, once you have splicing that's taken place, then these so-called exon junction complexes bind to each of the two exons when they come together, which is another mark that splicing has taken place. Yeah? So, yeah, well, so the question basically is what about HNRNPs? And so here, if you look at the HNRNPs again in the diagram, here's a couple of HNRNPs that should have been just associated with our introns, but there's still some of them here. And again, it has to do with the H part. Some of them are associated, some of them aren't. And there's no real obvious rhyme or reason to exactly what they are. Or I could give you an even worse set of alphabet soup, which we probably don't want to deal with, do we? No. Okay, so you've got the exon junction complexes. The other thing that, of course, you have is a cap, which associates with the cap binding complex we talked about before. It protects the end of your RNA from those hungry exonucleases that would love to chew in at the end. So you've got capping, exon junction complexes, and SR proteins. And then you need to have a poly A tail. How do you know you have a poly A tail? That's because you have all of these poly A binding proteins that are associated with it. And it's really much more about the proteins relative to the RNA. Because if you had only nuclear export taking place with some kind of specificity as far as the RNA is concerned, if you've got 20,000 genes, each of those which should be spliced up to 30,000 ways, you're never going to have enough ways of being able to say, okay, this RNA should go, this RNA shouldn't go. On the other hand, if you've got the proteins which are bound to it, you could recognize that those are the correct proteins that are only going to bind to a properly processed messenger RNA, and those then can get exported. And so it's the cap binding complex, loss of, I probably should have some HNRNPs here, knowing that splicing is completed because of the exon junction complexes, and the poly A tail is formed because of this poly A binding protein. These then get taken to the nuclear pore, which will transport this whole complex. So again, it's an RNP. It's your messenger RNA plus all these proteins that then get transported out into the cytoplasm, and we'll talk about translation next time. So what happens to, and I think somebody's asking about introns, not the introns that was left in, but what happens to exons that get left out, or some kind of RNA which doesn't undergo this whole process? It gets chopped up and recycled inside the nucleus. And what does that? It's what's called the exosome. Again, all these darn molecular biologists love to call their multi-macromolecule complexes zomes. This is multiple different exonucleases. Almost all of them are RNA exonucleases, which are going to chew in at 5' ends, chew 3' ends, and basically any extra RNA which is left around inside the nucleus, mostly going to be introns which are not being used, but also any other kinds of RNA. For instance, you have a mutation that means it doesn't get spliced properly. If that particular RNA is there, it will get chewed up by this exosome complex. So any more questions on messenger RNA? Everyone grabbing their clickers. I've got you well trained, don't I? Okay, so whether an mRNA is exported or not seems to depend on the presence of a cap, the presence of a poly A tail, the lack of introns, the presence of all possible exons, the absence of some mRNA binding proteins. This is not a great question. I probably should have rewritten it, but there's one best answer. Now, all of the above, F. I don't have an F on here. Five. 1. Okay, we have definitely a much more of a difference of opinion on this one. I am going to put the absence of some of those HNRNPs at the end. I think that's the best answer, but I'm going to keep thinking about this question because in reading it now, I think it's pretty unclear exactly what should be there. So the presence of a cap actually is going to be just the cap. It's not the cap-binding protein. And so the cap-binding protein will be associated with that. The same thing is true with poly A tails. Lack of introns, that's going to be something that is, again, is only being detected by the proteins. And it's by those proteins. So it's the absence of some of these proteins. So it's really all about the proteins. It's not about the RNA, which is what I was thinking of, but obviously I didn't do a particularly good job of putting that together. So I will think about whether I'm going to include this question or not later on. Yep, exactly. First question on the midterm. No, wouldn't do that. Oh yeah, sorry. Yeah, so the question here really has to do with what about export? And is it a sequence in the RNA which is important for export? Is it about the proteins which is important for export? And it's really all about the proteins. It's not about the RNA. So those proteins are going to get gone into the export machinery. So it's really a protein-protein interaction, not an RNA interaction. Yeah. Oh, so is the absence of some proteins, and those would be the HNRNP proteins, and some of the HNRNP proteins, but not the absence of all of them, which is, again, unfortunately rather confusing, but that's the terminology which is used. Yeah, so you could also say the presence of EJC, but if you have the exon junction complex, that's only going to tell you about one exon junction. And so if there are other exon junctions in there, because most of these have multiple exons in them, then you could have an exon junction, which is perfectly fine, but then another set which actually hasn't been properly spliced yet. Yeah. The answer is E, subject to modification, potentially. Okay, so today I wanted to finish up talking about one of those other kinds of RNAs. Who cares about these RNAs? It's because they're the most prevalent RNA inside a cell. And also absolutely critical for translation, as we'll see on Friday with all of our extra friends here. You'll probably not even notice. It's really all about the RNA. Translation is really all about the RNA. Mostly made by, as I mentioned at the beginning, RNA polymerase II. Doesn't have a CTD, so we're not capping, we're not tailing, we're not splicing any of these ribosomal RNAs. We need a whole bunch of them because ribosomes, most of the RNA inside the cell, we'll talk about next time, ribosomes are actually relatively inefficient, so you need a lot of ribosomal RNA. How do you get that? Usually it's by having many, many copies of your ribosomal RNA, about 200 of them in human cells, and you can actually see that if you look at actively transcribing DNA in human cells. The DNA is a little hard to see here. It's that little line in the middle. All of these sort of brush-like structures which are coming off of it, each of those is a ribosomal RNA transcript. So many, many, many copies of these. It's all made, I should say, the part that's made by RNA polymerase I is one really big RNA, also known as the 45S for Svedbergs. We'll talk more about Svedbergs right at the end of class. It's big, 13,000 nucleotides, way bigger than it actually ends up being at the end. Gets chopped, this particular piece gets chopped into 3 pieces. So it's not splicing. It's actually an endonuclease activity, which turns out to be U3. Everybody remember U3? Remember we had U1, U2, U4, 5, and 6? Where's U3? U3 is involved in ribosomal RNA processing. So it gets chopped up. The other thing which happens is this RNA gets heavily chemically modified. And we'll look at that in a little bit more detail in just a second. And there's also a small RNA here made elsewhere by RNA polymerase III. So what is doing all of these things? We already talked about U3, but there are a bunch of other RNAs that are involved in modifications here. And how are they different from the other small RNAs that we talked about? They're the SNO RNAs with an O as opposed to the SN RNAs. Why SNO? This is the small nucleolar RNAs. And it's the nucleolus where you have all of the ribosomal RNA modification that takes place. So what are the chemical modifications? There's actually a whole bunch of them, but we're just going to talk about two, which are the most common. They're pseudouridylation, which is literally taking uridine and flipping it over. So normally your glycosidic bond attaching to your sugar would be here. Now it's gotten flipped over. And then you have O-methylation. O-methylation should sound really familiar because we talked about it in caps. Some caps also are going to have methylation, which takes place there. So these happen throughout the ribosomal RNA, but only at very specific positions. So how does the modification machinery know where these specific positions are? Well, it turns out it knows because there's sequence specificity of all of these SNO RNAs, which can bind in a complementary fashion to parts of the ribosomal RNA that need to get modified. So there are about 200 of these, and it turns out that there are about 200 modification sites that happen in these large RNAs. So it's a complementary nucleotide binding. In this case, it's not the RNA that has the enzymatic activity. There are proteins, because now we have a SNO RNP, because of these proteins. These are the ones that have the enzymatic activity. So take uridine, flip it over. It's a methyltransferase, which will methylate this particular DNA. These two classes, again, the most common modifications, pseudouridine and 2'-O-methylation, are again ridiculously, wonderfully named box CD SNO RNAs and box HACA SNO RNAs. CD, box HACA. So boxes, which we talked about before, these are conserved sequences. These are conserved nucleotide sequences. So these are those conserved nucleotide sequences that you find in any of these. Are those conserved nucleotide sequences likely to be the ones that are binding to a specific place in the RNA? No, they're likely to be involved in binding to the protein. So CD box is binding to this methyltransferase, and HACA, which is a little bit easier to follow, the H stands for hairpin, and that's because all of these SNO RNAs have a hairpin right in the middle of them. And that hairpin structure is what interacts with the proteins, which are going to give you pseudouridine. There's also a completely conserved ACA sequence, which you also find in all of these SNO RNAs. So where are these things? They're all present in the nucleolus, also known as the RNP factory, mostly because we're putting together all the ribosomal RNA here and actually really assembling the whole ribosome, which is here. So we have transcription of our ribosomal RNA here. That then gets chopped up by U3, modified through the SNO RNPs, because the protein which does the modification, where it actually binds is going to be due to the sequence of each of those SNO RNAs. Ribosomal proteins get added here, all of it in the nucleolus, and then the individual subunits of the ribosome, which is now another RNP, get exported into the nucleus. These things are huge. It's really pretty amazing that they can get exported through nuclear pores in the first place. Other RNAs and RNP complexes like the telomerase, which again is a reverse transcriptase with its own RNA that's associated with it. Many of these things get put together in the nucleolus, and turns out all of the small nucleolar RNPs get put together in the nucleolus. So this is your ribonucleoprotein factory process. And I think, given that we've got 12 seconds left, we'll skip the last clicker question. Yes? Yes. You can take a look at it, but I'm not going to score it. So, which of the following determines, okay, we can score it? Yes? Okay. Which of the following determines the site specificity for pseudo-relation of RNA? U3 snRNA, HACA snRNA, CD-box snRNA, 5S RNA, the exosome. Make yourself lucky I don't lecture that fast. Once you've voted, you can leave. But then you want to know what the answer is, I know. 30 more seconds. $20 more seconds. 15. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Yes, HACAs. Sorry for keeping you an extra minute.